Well, hello and hello again. This is lecture 5 in the series on FDA, the fundamentals of U.S. drug regulation. I'm Dr. Tim Cote, the Professor of Regulatory Practice at the Keck Graduate Institute, and also the Chief Medical Officer at NORD, the National Organization of Rare Disorders. Today, we are going to talk about the drug biologic approval process. This is chapter 5 in the book, the Feldy book. But we're only going to go through half of it today. It is the meat of the course. It is the essence of what everybody wants to know who gets into regulatory affairs, which is how do I go about getting a drug FDA approval so that I can go and sell the thing and market it and make money out there in the world. So it is pretty darn central, and it's about 40 pages, so I've broken it into two halves. The syllabus says that it's the drug process once and the biologics process secondly. It's actually not going to be that way. The drug and the biologics processes are closely enough, very similar to each other, and so there are other divisions, though, of the material, including differences in exclusivities and incentives for development of particular drugs. So there will be a split in the chapter. It'll follow the chapter split, but it will not be split by drugs and biologics. So before a new drug or biologic can be marketed and sold, it has to be approved by the Food and Drug Administration. That's the whole essence of regulation, and that's why we're here. This is the core of what people want to know. We're going to consider new drugs, which include a number of different new drugs. New drugs are set up by the 1938 Food, Drug, and Cosmetic Act. Anything that came after 1938 is a, quote, new drug, okay? So new drugs include other things that are similar to new drugs, like abbreviated new drugs, over-the-counter new drugs. Over-the-counter drugs are all new, actually, because that classification came in the new period. New animal drugs, generic animal drugs, abbreviated new animal drugs, and biologics. All of those will be considered in this lecture and the next. So, a drug is defined under Section 201G1 of the Food, Drug, and Cosmetic Act as being one of four things. The first, to take history forward, is articles recognized by the compendia, things that are put in the USP or some of the other compendia that are included. Second definition is articles used for the diagnosis, cure, mitigation, treatment, or prevention of a disease. Those are all considered drugs. And articles other than food that affect the structure or function of the body. And lastly, articles for use as a component of the above. So food is excluded, but you should know that there is this gray zone area of things called medical foods. They are defined in the Orphan Drug Act, as so happens, because there are rare inborn errors of metabolism, such as phenylketonuria, in which people have to be on special diets, such as a diet which has a very restricted phenylalanine content. Those diets are essential medical products for the purposes of patients with rare diseases. They are what is commonly recognized as medical food, but there's not a very good definition of medical food. That causes a whole lot of problems for people with inborn errors of metabolism and their parents who have to, shell out tens of thousands of dollars for their special diet, which is every bit as essential as any drug might be, and yet if the reimbursement issues are enormous. It's just a real mess that we have over there on medical food. So I just want you to know there's this place between foods and drugs that is very complicated, and the agency really hasn't dealt with very well as yet. We can talk about that later. So, as I mentioned, a new drug is a drug that's come on the market after 1938, and most of the drugs today, actually, the vast majority of them came in after 1938. Some are held forward from antiquity, but most of them. Unless a drug is GRAS, okay, or generally recognized as safe, and this is important in the consideration of over-the-counter medications. We'll talk about that shortly. Well, or in a future lecture, actually. New drugs must have been approved, must have a approved NDA, New Drug Application, or one of its variants, such as an ANDA, abbreviated New Drug Application, which is what we talk about when we say generics. Generics have got a ANDA associated with them that's been approved in order to be legally marketed in the United States of America. So that's how it works. If you want to market your drug, you got to have either an approved NDA or one of the other variants, including a BLA. Okay, so the process components of how does one get a drug approved for marketing here in the United States. Preclinical investigation, clinical trials, your submission and the review of the NDA, the submission and the review, and post-marketing. So those are the four things that go into having an approved drug. We will not talk about post-marketing in this lecture. That will be into the next lecture, lecture 6, and many of the other components of what was included in chapter 5. So let's talk first of all, what does preclinical mean? It means specifically pre-human, okay, because the clinic is considered to be the part, even if you're testing it on healthy volunteers, that's considered clinical testing. Even though those people don't have a clinical disease, that's still clinical testing. So anything before humans is preclinical. So it's animal testing and laboratory testing, and that has two functions. The first is to identify the potential utility of the drug in treating a disease. And the second is to gather evidence that it's effective in humans. So you need to, that it's going to be safe to test in humans. So the first is gather some evidence that it's going to be safe and gather some evidence that it's going to be effective. And that is really, those two words, safe and effective, those are the essence of what you try to demonstrate in order to get your drug approved. So FDA notification of preclinical testing is not required. You can go out and test to your heart's delight in any animals you want, in any cells you want, do any kind of laboratory work you want, and you don't have to tell the FDA jack about it. However, when it comes down to the fact that you want to get to test these things in humans, you need to have done it in certain ways in order to make certain that you're going to be allowed to test it in humans. One of which is you need to have used GLP, which is good laboratory practice. And those are specified out in a whole series of regulations as well. Now, unapproved drugs that aren't approved, that are just going to be used for preclinical testing, can be shipped interstate from one state to another, to another, to another. If the drugs are properly labeled and shipping records are maintained. Although there are provisions in the law that FDA can shut down the interstate transport of drugs intended for preclinical testing if it decides that it's in the public interest, that something bad is getting out and actually harming people. There's some reason to do so. So, generally speaking, in the preclinical phase, there is not a lot of FDA involvement except in an anticipatory way. You got to do things right in the preclinical phase so that you're all ready to be approved to go into the clinical phase. But it's not like you have to submit and have your application reviewed. When that happens is when an IND is submitted, when you're about to submit an IND. And let me take a step back. Before you actually submit an IND, one is best advised to have what's called a pre-IND meeting. So we heard in an earlier presentation how to request a meeting. One of the first meetings that should be requested in any drug development activity is the pre-IND meeting. This is an optional meeting. FDA does not require you to have a pre-IND meeting. But if you do request it, you will be going forward with your protocol for your IND, with what you've done already in preparation for your IND to see if the FDA will decide that your stuff is safe enough to go forward. Maybe even talking about your broader drug development plan, and you will receive invaluable evidence from the very people upon whom your fate in pursuing this activity of trying to get your drug approved depends. So we at the agency, especially in orphan drugs, we're always strongly encouraging the pre-IND meetings. Okay, so you need to understand that the IND is a application for an exemption from the rule which says you got to have a licensed product in order to be able to go out and use it in human beings. So the focus is on safety, on the pharmacology studies, on the toxicology studies, making sure that GLP regulations were followed in the preclinical testing. This is Part 58, Title 21 of the CFR. By the way, I am not requiring anybody to memorize which parts and titles of the various laws, but I am requiring people to know which laws exist and what they pertain to out there. So those GLP regulations govern the appropriate lab work. The regs establish a minimum requirements, okay? Those regs, they subject the laboratories to inspection. So preclinical testing labs are subject to FDA inspection. If they want to, they can come in and check you out. They do provide penalties for non-compliance. They set minimum standards and structural safeguards, but they're not very specific, to tell you the truth. So for example, a regulation could state that the personnel engaged in preclinical testing need to be sufficiently trained to conduct the study. But it doesn't say what sufficiently trained is. Does that mean that you need to know how to run an NMR machine? Does it mean that you've had a high school or a college or a graduate school level diploma? Well, I mean, that is subject to interpretation along the way, and in some cases it's going to be very clear, and in others it may not be. It requires a separate quality assurance unit, which has to maintain copies of schedules and written protocols. But it doesn't say what those schedules or written protocols needs to be. The GLP requires a laboratory to be suitable in size and construction. So maybe your hallway closet is not suitable in size or construction, and it does not, however, state that you need to have a giant edifice with a phase 4 laboratory, level 4 laboratory in it either. It just says that it has to be suitable. So you get the sense here of what the regulations are setting up. They're loose, they're vague, they're not very specific, but at least they say you have to have something. So failure to comply may result in sanctions, and sponsors should be active in assuring that preclinical testing facilities actively comply with this. So that is when you are engaging all the work that you've done in a preclinical sense forward is when you put in your first IND, investigational new drug application. Well, the clinical testing is the most expensive part. Preclinical work is rather formulaic. People know pretty much what needs to be done. But when you get down to clinical testing, you're engaging the healthcare system itself. So clinical testing costs tend to track very closely along with healthcare costs in general, which, as you all know, have been going up through the roof for the past several decades. There is lots of FDA oversight here. So your IND application is your formal notification to FDA that your clinical trial is about to begin. And from the time that you've submitted your IND application, you've got 30 days. You have to submit your IND application at least 30 days before you intend to start your clinical trials. But the FDA can shut you down. They can give you a notification of what's called a clinical hold. So one of the misunderstandings in the community is people think that their INDs were approved. INDs are not approved. INDs are either stopped or they are not stopped. Okay, so an IND is a notification. It says, hello, I'm going to do this clinical trial. I'm getting started. And there are laws that say you have to notify in order to do the clinical trial. But then the FDA has the right, if they decide that this is a risk to patient population and patient health, to shut you down. And one of the reasons they might is because you didn't do your preclinical work satisfactorily, you didn't check all the boxes, you didn't do it under GLP, all kinds of reasons why one might decide that it is not allowed to go forward and that they give you a clinical hold. But you also need to understand that while your IND will naturally address questions of efficacy, your plan and your protocol to address that efficacy may bear little resemblance to, at the end of the day, when we come to the NDA, what the FDA requires to demonstrate efficacy. This is why a pre-IND meeting is so incredibly valuable and important, is that you need to know what they are thinking will satisfy your need to demonstrate efficacy. Because as you can imagine, all of us could develop all different kinds of protocols, but and all of them would have various strengths and weaknesses. If we had unlimited resources, we could do enormous protocols. But generally, there are not unlimited resources, and there are limited patients as well upon which to expend those resources. So it is wise to have a discussion as what would merit substantial evidence, remember, substantial evidence of efficacy for this product with the regulators. So clinical testing is the most expensive part. There's lots and lots of FDA oversight. The components of your IND are listed in the book. I think there are 8 components. This is nitty-gritty, nasty little itsy-bitsy stuff, but I hate to tell you this. It's sort of important. You got to follow the rules and follow the structure. If they say use a 3-ring binding and use tabs, okay? If they say, and they often do, in order to make it easier for the reviewers, make tab 4 be this, then make tab 4 be that. Okay, this is like there is a style book out there of tiny little rules about how to submit several different things, including the IND application, to the FDA, including some of the post-marketing adverse event reports. Follow these rules exactly, because you have to do it somehow, so why not do it their way and then not have any problems, okay? But the content is what's really important, and I think everybody recognizes that. So number 1 is the cover sheet, number 2 is the table of contents, number 3 is your introductory statement and general plan, 4 is the investigator's brochure, except for sponsor/investigators. When the sponsor, the person responsible for the investigation, for the drug's development, for the whole thing, is the same as the investigator, the actual person who's conducting the study. So there's often a disconnect. Remember, we have CMOs out there, CROs, excuse me. CMO is Chief Medical Officer. CROs are Contract Research Officers. Research organizations, CROs. And often large pharmaceutical companies will hire CROs to conduct research, either preclinical or clinical testing. So in those cases, the large pharmaceutical company could be the sponsor, but the actual investigators might be either a CRO or, for example, a bunch of academics at some hospital. Those would be the investigators. You need, number 5 is a comprehensive set of protocols. Protocols are the heart and the meat of what the study is going to be. 6 is information on the chemistry, manufacturing, and controls, often abbreviated as CMC. How do you make this stuff, and how do you have good control, that you have good purity and quality and non-adulteration of your product. Number 7 is the pharmacology and the pharm tox, toxicology information. Number 8 is any previous human experience, including human experience overseas. And number 9 is anything else that the FDA wants to know, they can ask you for. So you can see that the IND application includes 2 different kinds of pieces of relevant information. 1 is about the drug itself, tell me more about this substance, and 2 is about the clinical investigation. The pharm tox data has to support the safety of human testing. You have to affirmatively demonstrate that we have reason to believe from our animal studies that it will be safe to test in humans. And there's a whole collection of different studies, all the way going from very, very high dose studies, reproductive studies, mutagenesis studies, to it's very clearly stipulated. Whereas in the clinical testing, one has to make up the rules for how one's going to do the study all pretty much on yourself. So you have more of a blueprint for how to do preclinical testing. It's pretty much out there and standardized in a way that can be easily understood. Clinical testing, on the other hand, particularly when we're talking about orphan drugs or drugs for diseases that just aren't common, where that road has been trod 100 times before. In this case, we're trotting a new road. You often have to make up your own clinical protocol as you go along. So The protocols must be detailed with the names of the investigators, the inclusion and the exclusion criteria, the outcome measures, the monitoring responsibilities, and the application must include the IRB particulars. Now, we know what an IRB is, right? Institutional Review Board. This is the core of patient protection, and an an IND is an application in which you are trying to demonstrate to the regulators that you have sufficiently protected the patient by testing all this stuff before it's ever gotten to the clinic, and that you have this critical patient protection board in a place called an institutional review board. Now, there's a whole set of regulations that concern IRBs, and they are critical to the FDA. The patient participation has to be completely voluntary, and there has to be informed consent. The patient has to consent to say, yes, I'll do this, but it has to be informed consent. He or she has to understand clearly the risks and the benefits in advance of entering into the study. He has to be informed about the risks and the benefits and the alternatives, what other things they could possibly do. Subjects cannot be forced into the study, so they also can't be forced to waive their future claims against the investigators. So you cannot have a disclaimer within your informed consent saying, well, I agree that I'm not going to come after the investigators if something bad should happen to me. There are problems with children when one wants to do tests on children. Of course, their parents are considered the people who can give informed consent. But there's special problems with prisoners. The testing on prisoners is something that is usually avoided in this country, almost always avoided in this country. We have a history of that, and it's not a good history, I might add. Now, IRBs they're constituted by an institution. That's where the I part comes from. Usually it's an academic institution, a unit of higher learning. They have to be approved before the IRB has to approve the clinical trial before it begins, and it has to monitor it all the way to the end. So there's two functions. One is saying, yes, we're the Institutional Review Board, we've reviewed this, and we allow you to proceed. The other is, and we're going to continue it, and we're going to follow it going forward. IRBs have to be diversely constituted. They really need to include not only technical people who know something about the drug and the disease, but also members of the general community. Patients have to be involved. Sometimes a lawyer is involved. They have to include a collection of different kinds. Sometimes a priest is involved, actually. IRB records, the records of the IRB, are again subject to FDA inspection. So there's a lot of points in this process in which the FDA can walk in and say, give me your records. Okay, so clinical trials. We've said this is the most expensive part, and probably all of you are familiar with the classic taxonomy. The clinical trials are broken down into phase 0, phase 1, phase 2, phase 3, phase 4. Phase 0 is a relatively new concept. It says, before we even get into phase 1, let's test this with a tiny microdose of the drug in a human being before we get into fully testing drug safety in a bigger way. It's sort of more of a theoretical concern or a theoretical approach to drug development testing. I know of very few uses of it, to tell you the honest truth. I don't think it's very practical, but it is a concept in which trialists sometimes dabble. I myself need to learn more about it. It's a relatively new concept. The phase 0, you heard it here first, okay? We don't need to really say anything more about it. But phase 1 trials are classically small in number and usually or often healthy volunteers who've come forward in an attempt to assure that the drug is safe. So we're not looking to see, does this drug work in this clinical trial, test? Does it help cure the disease or anything? We're just seeing, are people going to drop dead once we start giving them this? And we need to collect pharmacologic data to create an appropriate phase 2. So things like blood levels might be collected in the context of a phase 1 trial. Now for toxic drugs, like some of the cancer drugs, which we know are inherently toxic, so there are ethical concerns about why are we going to pull people off the street and shoot them up with stuff that we know is going to harm them. If you know it's going to harm them, we wouldn't do that. So phase 1 trials for toxic drugs are frequently done in people with a disease to mitigate some of the ethical concerns about harming the healthy intentionally. So cancer drugs might be tested in cancer patients to begin with. Phase 1 is very few people. Phase 2 can extend up for mainstream drugs, can extend up to several hundred, and this is where your proof of concept comes in. Here's where you're treating people with a disease, and you're hoping to generate some preliminary evidence that the thing works, that it might actually be helpful in the disease. Phase 3 drugs are your definitive trials, also known as your pivotal trials. This is the real deal. This is the heart of your evidence that you are going to demonstrate to the FDA, look, my drug works. The Kiefhauser amendment has been satisfied. There is substantial evidence of efficacy using well-controlled clinical trials. Okay? There may be more than one phase 3 trial. In fact, the standard for mainstream drugs has traditionally been that there will be 2. Phase 4, so phase 1, 2, and 3, and then you submit your NDA, and presumably you get it approved. Phase 4 are post-marketing commitments. Okay, these are the studies that it can be that either the drug company wants to do themselves, not too likely, or that the FDA requires as a condition of approval, increasingly likely, that after the drug is approved, you must still collect this information, and it can be safety or efficacy. Safety is particularly important to the FDA, and phase 4 trials have traditionally had an emphasis on collecting additional safety data, but they can also collect efficacy data along the way as well. So the obligations for the investigator is to focus on patient safety and scientific integrity. The sponsor is responsible for informing the FDA of any safety-related events, adverse events. There are rules that say you're going to go out and you're going to conduct a clinical trial. Whenever something bad happens, you've got to tell the FDA. Okay? They're responsible for record keeping, for compliance with the regulations, and for being compliant with the IRB. They must report all adverse events. Okay. So, FDA, that's the Food and Drug Amendments Act of 2007, requires non-phase 1 clinical trials with serious diseases to be listed with clinicaltrials.gov. This is something new that you need to know. There was concern in the government that people with serious diseases, cancers and other really serious diseases, just didn't know how to get into clinical trials. They didn't know where to look. And FDA 2007 made it a requirement that trials need to now be listed so that people will know what the drug is, what it's for, and so on. And you should also know that people who are found to conduct clinical trials with a lack of scientific integrity, and this has happened a number of times. In fact, I will never forget, there was a large breast and colon study that had an issue of scientific misconduct that I was called in as part of the review team that involved probably 100 people at NIH reviewing documents that have been collected. You can, if you're found to have scientific misconduct, be disbarred from being able to conduct another clinical trial that's submitted to the FDA. Okay, so we have to move on through some of the rest of this material. The FDA oversight, what is the stick that it has? It can put things on clinical hold. There are lots of reasons for clinical hold. Those adverse events might be one of them, but the FDA may communicate that immediately with a telephone call. If so, it has to follow up in writing within 30 days, and it can also suspend or withdraw the IND if it decides that no, this isn't just a clinical hold, we're stopping this and we're stopping it for all time. So remember, phase 3 trials, the convention is 2 well-controlled studies. Okay, so hooray, hooray. You have finished your phase 3 trials conducted under IND status. You've done 2 or 3 of them. You know that your drug works. You're pretty sure that the risks, the safety adverse events are well known, and you have those well recorded. You're ready to prepare your NDA. And the NDA content includes, one, the preclinical data. So going back again, you still got to, again, support all that with preclinical data, the animal and laboratory studies. The farm tox information has to be included again in the NDA. Human pharmacokinetic and bioavailability data. Presumably, as you've been doing this IND work, you have collected blood and you have figured out the route of the transmission of the drug. You know how much of it is partitioned into which compartments of the human body, and how much of it is metabolized by the liver and how much by the kidneys, and so on and so forth. All that good farm tox stuff. And the pharmacokinetic stuff and bioavailability stuff. You know how much when you stick it in a pill and you put it in their stomach, how much gets into their body and into their blood. And so all that information needs to be included. You need clinical data demonstrating substantial evidence. That's the heart of your clinical trial. And your clinical trial outcomes, something we should talk about probably in a separate course on clinical trials, but they come into 2 main categories. One is clinically relevant outcomes, like the 6-minute walk test. How far can you walk? That might be a really good clinical outcome measure for something like, say, muscular dystrophy. If the people who were treated could walk significantly further than the ones who were not, that would be an important difference. Or there are these concepts of biomarkers, like what is your blood lipid level? And that is a whole lecture in and of itself, but the outcomes and the data that demonstrate that your outcomes are affected are all included in your NDA content. So 4, proposed methods of manufacture, processing and packaging. This is your CMC. How do we make this stuff? How are we planning to make this stuff? Where are we going to make this stuff? And under what kinds of conditions are we going to make this stuff? 5 is your drug substance description, the heart of the chemistry. 6 is a list of all patents or statements that there are no such patents. So the patent process is part of the NDA. And then 7 is the label that you're proposing. How are you going to label this thing? And is your label going to reflect properly the indication that you're recommending it for, the adverse events, the contraindications? How are you going to construct the label? And if we have some time, we should look at a label or two. I trust that most of you have seen a label, and if you haven't, let's talk about that. So, NDAs and INDs are secret until they are approved by the FDA. The FDA receives information from sponsors, which is considered commercially sensitive. So this is the way that the system is set up. The FDA cannot comment on even the existence of an IND unless the sponsor does so publicly themselves. And we already mentioned a little bit about clinicaltrials.gov, but that came in relatively recently, and it doesn't apply to absolutely everything, I don't believe. So you need to know that that the FDA operates under strict rules with regards to what kind of information it can release to the general public. I wanted to mention a little bit the REMS, the Risk Evaluation and Mitigation Strategy. It's specified under FDAA, so the Food and Drug Amendments Act of 2007, relatively recently. It says that the FDA may require, when it deems necessary to ensure a REMS, when it deems necessary to ensure that the benefits outweigh the risks. So this is a new way to approach drug safety. And it can include many different things. So it's, can the FDA require a program, let's call it, okay? A risk evaluation and mitigation strategy is sort of a mouthful, but it's saying empowering the FDA to require a program to limit risk. It sets out a timetable and periodic assessments. You can look at those in the text. But there are potential components. They include the potential components. This means in negotiation with the sponsor, in review of the data, in deciding what is necessary, the FDA could require any of these things. It could require a medication guide. It could require something special to the patient be included. It could require a healthcare communication. It could require that, oh, this drug can only be prescribed by a certain kind of physician, somebody with who has board certification in infectious disease, say, or a certain healthcare person. It could require patient testing. It could require, for example, oh, before you can prescribe this drug, you have to have the patient tested with beta HCG to make sure that she's not pregnant. Okay, that could be a requirement which is built in to the REMS, which is built into the approval process for the drug. So not just something that is adopted because it makes sense, but something that is required as a condition of approval. And generics must also comply with the REMS. FDA defines the REMS in an action letter, a letter that specifies out what it's all going to be. Okay, so a little bit more about the mechanics, and this is gone through in more detail in your text of how an NDA is processed at the FDA. When the NDA arrives, okay, you stick this thing in the mail. Sometimes you actually drive a truck full of all the paper. These days, more often, you send it electronically in a special file. But you stick the thing. Let's go back to sticking the thing in the mail. It gets there at the FDA. Hello. When it arrives there, it is received, but it is not filed. Okay. It is only filed when it is formally accepted for review. So there is a pre-screening process that before it's accepted and filed, it doesn't become a bona fide document until it's filed. It can be received, but it's not filed until it's accepted for review. The FDA has to file within 60 days of the receipt. It's required to do that, but it can refuse if the application is found to be incomplete or it fails to include the required certifications on preclinical and clinical trials. A refusal to file, or RTF, is usually for obvious screw-ups. The exception is for fast track under FDAMA permits review on a rolling basis. So under fast track, there are provisions for, okay, you can give me a little of this and a little of that and a little of that and a little of that. And then when it's all finished, then I'll look at it all. I'll look at it all along the way and maybe help you out with it as we go. But you still don't get an approval, a full-blown approval, until after everything is in and I've looked at it all. Just so you know, there's some considerations about this thing called progressive approval, which is a different concept, saying, oh, we'll give you a little approval and then a little more approval and a little more approval. It's being discussed right now in Congress and elsewhere. We can talk about that in some separate section, but it's nowhere near ready for prime time at this time. It may in the future, but at this time, generally speaking, your classic NDA, get it all done and get it all complete, and you hand it all in at once. But under fast track from FDAMA, you are permitted to supply material on a rolling basis. So FDA uses the refusal to file if you omit a section, or you have clear failure to include evidence of effectiveness. If that isn't clear, that there's some evidence. Or 3, if there's some omission of critical information. A pre-NDA meeting, have as many meetings with as nerve-wracking as they can be for sponsors. They are well advised to have as many meetings with the FDA as they can. And if the pre-NDA meeting is one such that could prevent a refusal to file. And if it's refused to file, the sponsor can request an informal explanatory meeting and can still request a filing over protest. But that's sort of like shooting yourself in the foot, because if they say they don't want to file it, why would you press the matter so hard? So there are substantive standards for the review approval. The basics are those 2 words I mentioned earlier, safe and effective. Those are the hallmark. You demonstrate that, you're golden. You don't demonstrate that, forget it. The 1938, as you know, had no efficacy requirement, but 1962, the amendment required it to be supported by the applications for a new drug to be supported by substantial evidence. It defined in law as evidence from adequate and well-controlled clinical studies. And by tradition, this has required 2 independent pivotal studies, both of which have an endpoint that can be demonstrated as different from a control by a value of p less than 0.05. That's another tradition. That's hardly a law, and there are multiple examples when that tradition has been ignored and drugs have been approved in spite of having two well-controlled trials, each of which reached an endpoint within a p-value of less than 0.05. FDAMA provides for one adequate and well-controlled study accompanied by confirmatory evidence. And there are multiple examples, particularly in orphan drug development, in which that was the standard that was applied that resulted in drug approval. Now, it may not be approved unless, quote, adequate tests by all methods reasonably applicable to show whether or not such a drug is safe for use, and so on. So there are clear rules that you also have to assure that adequate testing has been done and the thing has been shown to be safe. It's always, however, a balance. Every drug has risk. Every drug is not completely safe. The 1995 guidance that the FDA put out says, quote, FDA weighs the product's demonstrated effectiveness against its risks to determine whether the benefits outweigh the risks. Okay? Because disease has risks. Okay? Disease has lots of risks, like death from disease. So if your drug has lots of benefit, then the more benefit it has, the more risk will be tolerated. Because there are no absolutely safe drugs. So the point is that FDA does not say, oh, this can kill people or this can harm people, we won't approve it. The FDA takes the risks in the context of the benefits and makes that comparison. Good drugs get approved, presumably drugs that are more harmful than they're worth don't. That's the objective that we strive for as a society. So adequate manufacturing controls, however, are somewhat less negotiable or subject to deliberation. They are pretty much pro forma. You've got to make sure that you meet these. There's some things which are just plain expected, and the chemical manufacturing controls are in that category. GMPs must assure purity, quality, strength, identity, and bioavailability, and the label must meet the standards as well. I mentioned earlier the PREA, the Pediatric Research Equity Act of 2003. It can require that NDAs or BLAs include safety and efficacy for pediatrics as well, if it's required. It can be deferred for multiple reasons, being an orphan drug being one of them, by the way. But PREA was an effort in recognition of the fact that we don't have good data on drugs for children, and that we need that in order to properly serve our pediatric population. Now, usually when it's a new drug, something innovative, something groundbreaking, this is going to come up before that advisory committee review that I mentioned earlier. The advisory committee can include consumer interest groups, patient representatives. The advisory committees generally respond to specific questions. This is the structure of which the advisory meetings go, is that the committee, assembled by the FDA, will respond to questions posed to them by the FDA, and one of which will be, is there evidence of efficacy? Is there evidence of safety? Those might be classic kinds of questions to put before an advisory committee. The agency is likely to use an advisory committee if it's new. The FDA must make a final decision on the matter reviewed by the advisory committee within 90 days after the advisory committee makes a recommendation on it. So that's when the clock starts ticking and people wait to see, is the FDA going to accept or take off or not accept what the advisory committee recommended. You should know that the advisory committee votes. They have to vote simultaneously. Sometimes their votes are close, okay? They don't always agree. So if the advisory committee comes out with a 4-5 decision that this is both safe and efficacious, then the agency has a lot more leeway than if they had come out with a 10-1 decision that it's efficacious and safe. Okay. Before July 2008, there was a system of approved slash approvable slash non-approvable. That's confusing, right? So they made a new system in July 2008, and they combined the approvable. So approved means, okay, go forward, sell your drug, this is great stuff, we want this for the American people. Approvable means, maybe it will be, but we don't know about some of this stuff. It's not quite adequate yet. You got to do some more work, and you got to go out and get some stuff. And then non-approved means, non-approvable means, no, this is junk, we are not allowing this. To tell you the truth, many decisions came into the approvable category. Now they've changed the terminology, and it's approved or it has a, quote, complete response letter. That's just FDA speak, but you need to know what it is because people will talk about, oh, I got another complete response letter. Well, what's that mean? It means that no, you are not approved. Okay, you were not approved, and then it says why you were not approved for marketing, approved for marketing. So the Food, Drug, and Cosmetic Act specifies a 180-day limit, but the FDA usually requires longer. The standard time is about 10 months to get a full NDA review. There are some interesting gymnastics and clock tinkering, which are described in your text. I found them a little legalistic, and I didn't really want to go absorb all this amount of time here today on that. But PDUFA has had a stronger influence on the performance goals, and those performance goals that PDUFA stipulates have actually affected some workflow changes at the FDA, since now it's being properly resourced to do those reviews. There are resubmissions for an NDA if it's not approved. Class 1s are minor updates. Say you didn't get the label quite right, and so you can tinker with that and fix it. That's a Class 1 submission. And Class 2s are anything else. Okay, so this was your first lecture on the drug and biologics review process. Biologics are also very similar. We'll talk about a few differences next week. Next lecture. And the second half will deal with the second half of Chapter 5. Thank you so much, and we'll talk to you again very soon. Take care. Bye-bye.